Welcome back, my people. How are you, my people? How are you doing? So today I'm gonna take you through something very interesting, something very simple. And guess what I'm talking about? The staining techniques. We need to understand what is a stain. It's just a dye used to add a color to a tissue, microorganism, or a cell for easy visibility. Basic, very simple thing. And you know there are different stains that are used in the laboratory, but today I'm gonna discuss with you a very crucial stain that is used in the laboratory. I'm talking about gram staining. So make sure you save this video for future reference, and also make sure you click to that plus button so that we can get more and more tips about this. And also make sure you follow at my YouTube channel there at microbiologist254, Instagram at microbiologist254, also on Facebook at microbiologist254, verified account. Let's dive in. So a good example, I'm gonna give you a different types of stains we use in the laboratory. We have the Romanowski stains, we have the Wright stain, we have the Leishman stain, we have the James stain, and we also have the Pap stain, and also the hematoxylin and eosin stains. These are just some of the stains that are used in the laboratory. But we have one very important stain that is used in the microbiology, which is the gram stain. So gram stain, I'm gonna give you a quick and a basic way to understand this gram staining technique that is used in the microbiology. So gram staining is just a basic technique used in the laboratory to differentiate between two groups of bacteria. I'm talking about gram negatives and gram positive bacteria. So they differentiate these two organisms based on their color change, that is the pink in color and the purple in color. So gram positive bacteria, they're able to obtain that or to get that purple color, and gram negative bacteria, they're able to get that counter stain that is red in color to produce that pink in color. So gram staining, why are they gram positive purple in color and why are gram negative pink in color? Basically, we are talking about one thing which is very important to a cell of a bacteria, that is a peptidoglycan membrane. So peptidoglycan membrane, the gram positive bacteria have thick peptidoglycan membrane, while the gram negative bacteria, they have a thin peptidoglycan membrane. So we have different substances or different reagents that are used for these gram staining techniques. And guess we have four of these reagents. I'm talking about 1, crystal violet, 2, Lugol's iodine, 3, decolorizer, and the other one, which is a secondary stain, which is a counter stain or a secondary stain, that is safranin. We have a basic way to understand this, and I usually use the formula, that is, it is CIAN, C-I-A-N. C for crystal violet, I for iodine, which is Lugol's iodine, A for acetone, which is a decolorizer, and N for neutral red, that is the counter stain. So what about the time that is used? I usually say it is 11 to 01. 11 to 01, that is 1 minute, 20 seconds for the decolorizer, and 1 minute for this counter stain, that is the safranin or the neutral red. Despite that, the procedure is very simple. You just prepare your smear, you fix it by heat. After that, you do what? You flood the crystal violet on your smear for 1 minute. You let it stand for 1 minute. After that, you wash, then you tap the slide. Then later, you do what? You add Lugol's iodine for also 1 minute. That is a mordant. After that, after 1 minute, you wash the smear. Then you tap to remove the excess water. Then you do what? You add the decolorizer. Decolorizer, that is acetone, just for 20 seconds. Depending on the company, there's a company that says 15 to 20, or the other company that say up to 30 seconds, but minimally you can do it with 20 seconds. After that, you wash, then you tap to remove the excess water. Then later, you do what? You add the counter stain, which is the safranin. Safranin also, you allow it to stand for 1 minute. Then you wash your slide. After that, what you do? You let the slide to do what? To air dry. Then you will observe what? The color change. Of course, we have two things. Either it is pink or it is purple. Purple for gram positive. Pink for what? Gram negative. After that, you'll get to differentiate the different morphological structures of these cells. We have gram positive rods, gram positive cocci, gram positive diplococci. That is among one of the morphological about the characteristic of gram stain. The other one, we can have a gram negative rods, gram negative cocci. We can have the gram negative diplococci. So those are just the other one about the gram negative. But basically, we differentiate two groups of microorganisms, that is bacteria, that is gram positives and the gram negative bacteria. Tips. Basic formula, it is just CIN, CIN, cyan, CIN. C for crystal violet, I for Lugol's iodine, A for acetone, N for neutral red. But remember, we have an alternative. If you don't have the crystal violet, you can use what? Gentine violet. That is another alternative. And for the secondary stain, which is neutral red, if you don't have the neutral red, you can use what? The safranin. So, but the procedure remains as it is for the other staining. So, basically that is just simple and quick one about the gram staining. Understand tips? Used to differentiate between gram positive and gram negative. Period. Thank you guys. Make sure you like, make sure you save this video, and also make sure you share to many people to get to understand this basic about the gram staining technique. Thank you.